Assalamualaikum and salam sejahtera. My name is Muhammad ZAKWAN ZAKIRIN bin Abdul Razak. In this video, me and my team members, which is AMZAR, RAZIQ, Muhammad DANISH and Shazly Iman, will present about the shingles disease. So let's start with the presentation. Now let's start with the introduction of the shingles. Shingles is a painful rash that is brought from the viral virus, and it will cause a skin disease. This shingles can happen in any parts of our body, but it is typically manifest as a single band of blister that wrap around chest, either it is left or the right side of the chest. So, as we can see in the figure, this is the shingles skin disease. Shingles is caused by the a virus that named varicella zoster virus. That is also can be known in scientific name, which is human alpha herpes virus 3. This virus, a species of dsDNA, that come from the family Herpesviridae. This varicella zoster virus that also caused the chickenpox. And after we get the chickenpox, this virus is inactive in the nerve in our close to our spinal cord and brain, and this virus will resurface year later as shingles. So for this is for the diagram, this is the varicella zoster virus genome. So we have a wall which is a lipid envelope, and outside the wall we have the glycoprotein spike, and inside the wall we have tegumen, nucleocapsid, and double-stranded DNA which is inside it. So this is how the varicella zoster virus can be affect to the human, which is the virus is we inhale the virus and it will move to inside our nerve fiber, and after that it will go to our subcutaneous layer, nerve fiber, dermis, epidermis, and lastly it will affect in our skin or lesion. This virus, it is not a life-threatening illness, but it can be painful and rashness, which is always on common skin disease. To preventing shingles, a vaccine that called Shingrix can be a way to prevent the shingles. From the Center of Disease Control and Prevention, which is CDC, recommend Shingrix for adults 50 years and older to prevent the shingles and related complications. Shingrix is not a non-living vaccine made of the virus component, and this vaccine has been shown to reduce the severity and duration of the disease. It also can lower the risk of the postherpetic neuralgia, shingles complication, which is the shingles pain, and also the blister also can be healed. And after that, this is the varicella zoster virus DNA sequence, which we get from the NCBI website. I think that's all from the introduction. Next, we move to the method of employment. Okay, thank you.
Okay, Bismillahirrahmanirrahim. My name is Shazly bin FANSURY, and today I'm going to talk about my project of cellular and biomolecular project, which is about creating a vaccine, which is in our group we make on about shingles. So as we can see that the first method of employing is extraction. So what is extraction? Extraction is a method to purify a DNA by using physical or also chemical substances for separating the DNA from cell membranes, proteins, and other cellular components. The main purpose of the DNA extraction, or we can say that DNA isolation, is to provide a new or a pure DNA, a DNA that's been evolved into a new cells. So as you can see the figure below, we can see that the first one on the right side is muscles has been injected with RAZAK, which means uh, zoster vaccine, recombination zoster vaccine, which is it includes AS01, which is stands for adjuvant system, and also GE, which is glycoprotein, and the first one is which is the muscle, and also next is the circulation tissue, uh, which consists of glycoprotein with specific antibodies and also glycoprotein with specific T cells. So as you can see the figure, uh, it shows that the CDT cells and also specific B cells, which is a lympho node in the circulation below. And below you can see there's a types of cells, which is dendritic cell, macrophage, neutrophil, B cell, and CD4 plus T cell. So the first step is any nucleic acid purification reaction is releasing the DNA RNA into a solution. Lipid nanoparticles, also known as LNP vaccines, were made using modified mRNA procedure. Lipids were dissolved in ethanol, which means it dissolves in acid amino. So the next step is extracellular lipids were mixed with 100 millimeters citrate buffer with a uh, with a scale of pH 4.0, which is uh, which is acid, which because it's below 7. So the next step is step 3, which is um, a single zoster vaccine glycoprotein and adjuvant system stimulates the zoster vaccine specifies antibody and also CD4 plus T cell responses in high risk population. Synergistic actions of the vaccine adjuvant system driven the vaccine to make the immune response, including the polyfunction T cells. And the next step is the circulation tissue on glycoprotein specific antibodies and the glycoprotein on specific T cells. And the next part I'm going to pass to my friend. Thank you very much.
For the PCR amplification, the polymerase chain reaction, which is also known as PCR, is a simple technique that amplify a DNA template in vitro to produce specific DNA fragments. This method can detect more specific sequences more sensitively and achieve higher level of amplification in less time than previous method, which is the traditional method of cloning the DNA sequence. Before proceeding to PCR, it need to fulfill the requirement, which is we need a DNA sample for the varicella zoster virus and then a primer DNA polymerase, and also the nucleotide solution mix, which contain the adenine, uh, thymidine, cytosine, and guanine. So for the PCR, it has 3 steps. So for the first step, which is heat denaturation, which heating the original sample hydrogen at least 94 degrees Celsius uh, to help the hydrogen bond to dissolve and separating the DNA into single strand. After that, which is the step 2, which is primer annealing, which is cooling the sample mixture into the temperature of 50 to 60 degrees Celsius, and the DNA primer and DNA polymerase enzyme can attach to the separate DNA strand. Then the DNA strand have been separated as the result of heating, and the nucleotide of the ATCG from the additional mixed solution will pair with them. And lastly is the primer extension, which is the combining the solution and the single strand DNA to create a new complementary strand of DNA. So from each of the single strand, it can duplicate into a double-stranded DNA molecule. This process can be repeated approximately 35 to 40 times, and every time or cycle perform a heating and cooling cycle, and the resulting of DNA sequence is also can be double. And after like 35 doubling cycle, a single short piece of DNA from one sample can be amplified to generate millions of copies. So, as we can see in the figures, this is the first cycle, which is the denaturation, and after that is the annealing, and lastly the extension. For the second and third cycle, we can repeat the step again until we make until 35 cycle. That's it for the PCR amplification. Thank you.
Assalamualaikum, my name is AMZAR and I would like to continue in the report, which is the third point, purification of DNA. So there are 2 standard techniques that will be employed in the purification process, which are centrifugation and chemical extraction. So what is centrifugation? Centrifugation is a method that separate the molecules that have different densities by spinning them in a solution using a centrifugation machine in high speed. And chemical extraction is to extract the centrifugated molecules and extract it further. So the first step in the process is the sample is centrifuged at high speed to cause the heavier components to settle to the tube's bottom, which means to separate the molecules. Then the unwanted substances that are still in the solution are eliminated. Then the water is used to redissolve the precipitate in the tube. Then to dissolve and denature the protein, phenol is added to the tube. Phenol is one of the most common use of chemical extraction. Then the 2 solution layers are created. Water makes up at the top layer and phenol at the bottom. After the mixture has been mixed, the protein will have dissolved in the phenol. Then the water containing DNA and RNA is drawn out and placed in the different test tube. RNA is broken down into tiny fragments by the addition of the enzyme RN ribonuclease RNase to the test tube. Then the test tube is filled with alcohol. Small RNA fragment will continue to be dissolved in the solution as it uses up all the water and injects the larger, less soluble DNA out of the solution. Then number 10, the DNA settles to the bottom of the test tube after another round of centrifugation. Then the RNA fragment containing solution is drained out. And lastly, buffer is used to restore the tiny DNA pellets. Then I'll continue to the fourth point, which is the separation of the DNA. So electrophoresis is used in the separation of the DNA. What is electrophoresis? It is an electrokinetic process which separates charged particles in a fluid using a field of electrical charge. Depending on the type and size of the molecules, it can be accomplished using a variety of techniques. In the life science, it is more frequently employed to separate DNA or protein molecules. While the processes vary in certain aspects, they all require an electrical charge source, a support medium, and a buffer solution. In lab, electrophoresis is used to separate molecules according to their size, density, and purity. Electrophoresis works when molecules are exposed to an electric field. A force acts upon them since the molecules are electrically charged themselves. The electrical field generates more force on molecules with higher charges, which cause them to travel to the support media at a distance proportional to their mass. So this is the figure for the electrophoresis process. Then agarose is one of the common gel to use in the electrophoresis process, which is a form of electrophoresis used for the separation of nucleic acid fragments based on their size. When electrical current is supplied to an agarose gel, negatively charged DNA or RNA migrates through the pores in the direction of the positively charged end of the gel, with smaller fragments migrating more quickly. Ultraviolet light can then be used to see the resulting bands. Once the DNA fragments was visible, the DNA band was compared to the DNA ladder before being cut off the gel, which contained the DNA we sought to extract. That's it for me. Thank you.
Continue with choosing a vector. So we need a plasmid DNA, 200 ng extract DNA, 5 UL water, 15 UL master mix, upstream and downstream. So actually the recession enzyme have to cut the plasmid vector for the PCR amplification. The plasmid vector in this instance is PUC19. Plasmid vector PCR amplification follows the same denaturation, annealing and extension step as gene PCR amplification. So the conclusion is vaccine is to prime the immune response, so the immune memory can facilitate a rapid response to educate, control the pathogen on natural infection and prevent disease manifestation. It provides for the development of safe and effective vaccine. So Shingrix is the first novel herpes zoster vaccine to be licensed by the FDA in almost a decade, making it the first vaccination of its kind ever. The Advisory Committee on Immunization Practice (IACIP) recommend vaccination with Shingrix for healthy adults aged 15 years, including adults who have previously received Sotavac, in order to prevent herpes zoster and associated complications. This recommendation is based on Shingrix efficacy as well as its safety. So that's all from us. Thank you.